Good day everyone, my name is Biotechnology. Today I am going to report rust resistant wheat and soybeans. So before that, first we'll know what is rust. Rust is a fungal disease caused by various species of fungi including Pocinea, Phacopsora, and others. So how does rust spread? Through spores and infected plant materials. So rust symptoms vary depending on the types of rust and host plants, but common signs includes orange or yellow spores, lesion, chlorosis, and defoliation. So rust has a negative effect. First is reducing photosynthesis, disrupted nutrients uptake, increased water loss, reduced yield and quality, impact on plants' hormones, increased susceptibility to other disease. So now we know what is rust and how rust affect to a crop if the crop will be infected. So now, let us proceed to the rust-resistant wheat. Wheat is one of the leading food crops around the world. It provides roughly 20% of calories and protein for human nutrition globally, staple for billions, particularly in regions like South Asia, North Africa, and Middle East. Wheat is a staple food of the many countries. So through this, because it is their staple food, so they plant it in large scales. And the bad side if we plant in the large scale, because it is very susceptible of infestation, because it is a large area that easy to be infected. There are three types of rust that infect our wheat or infect wheat. First is striped yellow rust, Ponesia striformis, sp. cause yellow stripe on leaves. So particularly, it can be seen in the leaves of our wheat. Number two is leaf rust, Punesia triticina, orange-brown pustules on leaves, reduces photosynthesis. It can be seen also in the wheat leaves, but in orange-brown color, and it will reduces the photosynthesis, because we all know that the area where the photosynthesis happens is in the leaves. So the rust in the leaves will penetrate to the cells, and that cells, if the plant were know that it will infected, the plant will sacrifice that cells in order to the fungi not spread uh, in the wheat. Number 3, stem rust Funesia graminis sp. Triticea forms red brown pustules on stem, most destructive. The stem is like a highway of every nutrient comes from the roots that that the nutrients will be taken. So because of this, there is an economic loss. Stem rust alone can cause global losses of 6.20 million metric tons, which is valued at USD 1.12 billion annually. And it reduces food production, or it limits, or it threatens the food security. Rust poses 10 million people into hunger. So now we are tackling about the life cycle of our rust, wheat rust. So first, it undergoes your spores, teliospores, basidospores, spermatia, and ascospores. So now I have a video here uh, talking about wheat rust life cycle. So listen and understand the concept why rust, wheat rust, wheat rust is very destructive and very difficult to control, because it undergoes a lot of mutation. It can survive different condition. So you can see it to the video. So please listen. So why fungicides aren't the answer? First, the cost. Because fungicide costs $30 to $50 per hectare. It is unaffordable for 80% of smallholder farmers. Number 2, resistance. Rust evolves to avoid chemical in 2 to 3 years. So it is the very pain point of this uh, why they are not using fungicide, because because wheat rust resistant evolves, and when the wheat rust evolves, it is another problem that our expert will be solve. And also the very important environmental harm. We are not only killing our target host, but also our beneficial insect, our pollinators, and even can harm to us. And here comes the genetic resistance. Experts have identified that the most durable, effective, and environmentally sound strategy to combat wheat rust is through genetic resistance. Two Big Foundation and partners developed the first genetically engineered wheat lines by sucking 5 resistant genes. Thus, seedling assays in the greenhouse and adult plant tests is in the field have shown that the genetically engineered wheat plants remains unaffected even under high disease pressure, thus preventing outbreaks efficiently. Border-based resistance to the most virulent stem, Raspanin isolated, was exhibited at the seedling stage. So through the genetic resistance, they say to their um, trials or testing that is really effective to combat rust and can be uh, resist different kind of rust. So Cereus resistant gene, the ESR versus EPR. The ESR or all state resistant genes provide strong protection from specific type of rust fungi during all developmental stage. Limitation: if only a single gene is deployed to the plant, the fungi can evolve to be stronger and overcome the plant protection. So all state resistant is, or ESR is, the target is specific type of rust. So if you are targeting a specific rust, so there is possible that uh, the other kind of rust will be not be resolved because you are only focusing on that one. And then use single ESR genes lead to successive boom and bust cycle of persistent breeding against all three rust type in wheat. So probably in the first place, it can give you, the ESR give you good resistance to your wheat crops, but through the time, through the years, it will be um, overcome by the fungi because fungi will evolve to the certain specific resistant gene that you have employed to your crops. That is the reason that time comes, because you are using only specific genes to your crop, there will possible mutation, and through the years it will overcome your crop. So adult plant resistant or the APR genes, APR can confer longer resistance without developing pathogen evolution. Some APRs can also provide broad spectrum resistance to all rust fungi races, and in some cases to multiple rust species and other pathogen. So the adult plant resistance is not only focusing on the specific but on the broad spectrum of the rust that may be will affect to the weight. And then the EPR has a multiple rust resistant rust species that um, can be resist because it has a broad spectrum of resistance. So combining the ASR and APR genes can provide a more effective and long-lasting resistance in commercial available wheat varieties. So there is a advantage and disadvantage of the two. But when you combine it, there is possible that you can produce or you can create a very resistant uh, genes that you can transgene to your uh, desired host that will be able to resist different or many kinds of rust. These two kind of cereal rust resistant gene came from the conventional breeding, and the conventional breeding has challenges because it takes 8 to 10 years to stock 4 plus resistant genes, and then genes scattered in offspring losing efficacy. So it will scattered when you breed or when you are doing conventional breeding. The desirable genes will be scattered and not placed on the specific area. Then wild-weight relatives have resistant genes, but transferring them is rare, 1 in 10,000 plants. So because of conventional breeding, you are requiring to get numbers of resistant crops to be to crossbreed to your plants to get the desirable uh, or to get multi-genes. Stock for stronger resistant. Scientists have been invented a way to stack 5 plus resistant genes into a single cassette like a DNA toolbox. 100% of the offspring retain resistant, avoiding junk DNA from wild relatives that reduces yield. So, in this case, our scientists have come to a bright idea using genetic engineering, using biotechnology to stack 5 desirable resistant genes that can be resist weight rust. It has an ace to find a resistant gene or to acquire or to replace it to another crop because of this kind of idea. So now you can see the picture below, the traditional breeding or conventional breeding versus modern techniques. You can see that the traditional plant breeding, the undesirable genes and the desirable genes uh, crossbred to a commercial variety. Then you can see that the random multi-genes transfer, so it not in place when it in a new plant variety. On the other hand, the modification using biotechnology or the modern techniques, the desirable traits will be collected and then transferred to the commercial variety. And then in the new plants variety, the genes is located on the specific area. It is not scattered. That is the beauty of biotechnology or genetic engineering. Big 5 weight lines, proof of concept. The two Bleeds Foundation and its partners conducted a proof of concept study that showed that the use of multi-gene cassettes is an effective strategy to boost wheat resistant to stem rust. 5 stem rust resistant gene in a transgene cassette was introduced to breed weight as a single locus. So you can see the diagram. So the 3-5 is high. It is at the host plant where the 5 resistant genes is transferred through biotechnology. So the yellow color or the brown color is the stem rust specific ESR all-stage resistant gene, and the blue color is the multi-pathogen or EPR genes. Since the 5-gene cassette also contains the multi-pathogen EPR gene LR65, 67, the big 5 weight lines were also checked for leaf rust resistant, assays showed up to 39% less infection compared to the wild type control. So we all know that the big 5 weight is focusing on only on the stem rust, because we all know that the very disruptive kind of rust is the stem rust. So but the expert or the scientist also examine the big 5 cassette, its efficacy in the leaf rust resistant, and it shows 39 less infection. And also, in the stripe rust studies evaluating stripe rust resistance from YR46 is currently in progress. Field test 2018 to 2020 big 5 weight lines have near complete immunity to a US stem risk or QTG HGC. So the stripe rust is already completed, but still in its ongoing process to well develop its resistance to stripe rust. Cloning strategy, iterative gateway recombinase technology. Gene cassettes are usually compact, carrying a single gene and recombinase site. Thus, with the 5 large size resistant gene, it is challenges to develop a multi-gene cassette. So, our scientists came up in the cloning strategy because of the large size of our resistant gene were located on the specific recombination site, and it is challenges to develop a multi-gene cassette. That is why we came out with the idea to cloning method, iterative gateway recombinase cloning strategy that uses accessible lag selection for large construct generation. So we can see in the diagram that there is a donor vector attached to the gateway recombination destination binary vectors. So the lag Z and R2 is attached to the R1. After that, after the lag Z and R2 attached, the lag Z were removed and the lag Z were bind to R3, and then the lag Z and R3 were again bind to R2 and R1. Methods of cloning: highly effective without need for restriction enzyme, site availability or sequence domestication. Generate inserts that are more than 60 kb in size, containing up to 8 resistant genes. So it is the advantage of cloning. We are not restrict the enzyme site availability or sequence domestication. So you are just copying cloning to non-GMO status, removing selectable markers via gene editing, qualifiers under US secure rules. So non-GMO status, removing selectable markers via gene editing. So the difference of gene editing and genetic engineering is gene editing can be non-GMO if you are not transferring genes to another host. You are just edit the genes where is present to that specific crop, and then stock more traits to ensure robust and lasting protection against the destructive with pathogen evolution for years on or even decades. Number 3, breeding flexibility. Stacking genes in a single locus would allow continuous breeding for vital traits such as resistant to biotic stresses, as well as yielding height or milling properties. And then there is a global partnership or impact the Tublis Foundation and the consortiums of global partners including Cicero and the University of Minnesota. This partnership or foundation organization will unite to find solution to the wheat rust. And then semi collaboration working in the and introduce resistant genes in the semi varieties and to distribute resistant lines to their breeding channels. Soybean resistance, addressing the threats of Asian soybeans rust. The value of soybeans, versatile crops with a high economic value, climate tolerance, high protein and oil content, improved soil nitrogen, cash crops for smallholder farmers due to higher prices. So we all know that soybeans is very versatile, and it is also legumes that can improve our soils, and some of smallholder farmers make it as a cash crop because it for its high price. In the Africa, there is a large-scale farm where soybeans planted. Meeting the growing demand for soybeans. Soybeans demand in sub-Saharan Africa projected to more than double by 2050. African contributes only 1% of global soybeans production, heavy dependence on costly imports. So the needs of sub-Saharan Africa is possible increase or double by 2050. And because of this, Africa contributes only 1% of global soybeans production. So because of the increased demand of soybeans, they are tend to import that is costly. That is why they increase domestication production to reduce costly imports and bolster local economies. Many African countries have sought to increase their domestic production. So the African country came to an idea to increase the domestication production, creating policies promoting agricultural innovation. With soybeans promising a higher price than other staple crops, smallholder farmers have increasingly embraced greater production. So all of the people, smallholder farmers in African countries were increased in the domestication production of soybeans. And then the emerging of trade Asian soybeans. Because of the large-scale farming practices of soybeans in the African country area, there is the large possibility that it will susceptible for infection. I give you an analogy. If your target is huge, it is easy to hit, but if it is small, it is hard to hit. It is just like the situation in Africa, because most of the farmer increase domestication production of soybeans, so there is the possibility to be infected or to be disrupted of a disease, particularly rust, which is easy to spread. And then the fast-moving aggressive disease caused by the airborne fungus Pachypsora fakiesrizi can destroy up to 90% of soybeans crop within 3 weeks. So there is an emerging threat of Asian soybeans rust, and in just 3 weeks, it can destroy 90% of soybeans crop, leading cause of yield loss in Africa. Because of this crisis, they are leading cause of yield loss in Africa. Then combating SR or Asian soybeans rust challenges with fungi. Fungi can control SR but are expensive, potentially negative impact on human health and the environment, rapidly developing tolerance to chemical controls. So we all know that fungicide is effective but are costly, and there is a negative impact on health, and also the excessive use of fungicide can lead to the mutation or evolution of our rust, and it will again another problem to be solved of our expert. That is why it is not advisable to use fungicide. That they are use traditional breeding and screening effort, searching for resistant genes within soybeans for breeding. Because it is traditional, it is hard because you are need a lot of plants, thousands of plants to be screened if it is carrying a desirable genes that can be used to combat or to resist rust. Limited success as resistant genes are overcome by pathogens. And it is on trial and error. And it is the reason why it is hard, because trial and error. And it takes time. And you are not sure if your genes that had been gathered or yet will be effective. Time consuming and potentially inadequate. It is time consuming, like the wheat rust resistant gene. It takes a years, 5 years plus to get 5 plus rust resistant gene. That is the bad side of or disadvantage of traditional breeding. Extensive field trials by USAID soybeans innovation labs. Few lines showed tolerance but none with a robust stand. It is a very hard in traditional breeding that you are not get the standard resistancy of your crop, because it is trial and error, and you cannot really know what would be the possessed gene of that crop. And then comes the ideas of calling on the relatives, the pigeon pea solution. Exploring resistant genes in related legume species. So the soybeans has a relative species, legume species, the pigeon pea. Successfully transfer on the pigeon pea genes, the CRPP1 into soy, strong resistant to SR achieved. So finally, our experts have found resistant genes to our Asian soybeans rust, which is in a pigeon pea, the CCRP-PP1, and it can be disrupt or resist the rust. Because of the biotechnology, it is possible. Advantage of biotic approach: broad pool of resistant genes from crop relatives. So there is a pool of resistant genes from crop relatives. So it is easy to find and to transfer resistant gene into a host that you want to be applied. And then genes can be introduced into high-performing varieties, stacking genes for greater durability or persistence. Delivering is resistant soybeans, improving yield and income for farmers, reducing exposure to chemical sprays, more knowledge for researchers, increase access to a rich source of proteins and nutrients to consumers. Conclusion: genetic resistance powered by biotechnologies and advanced gene transfer is a vital for safeguarding global food security against devastating crop diseases like wheat rust and Asian soybeans rust. This approach promises resilient yields, enhanced farmer livelihoods, and a sustainable, nutritious food future for billions worldwide.